I'm very proud to introduce our first speaker, Shin. Jay's a group leader at Riken, Japan, and we worked together a long time ago. And he's also one of the godfathers of HCA Asia. So, Jay, go for it.
Okay. Good morning, good afternoon, good evening, everyone. It is a great pleasure to kick off our 5th Human Cell Atlas Asia meeting. And thanks, Alistair, for hosting us, and to Australia for this event. And very excited about some of the current activities that have been going on, not only where I work at Riken, but also broadly across Asia and globally in HCA, in the international community. But I just thought that would open up today's meeting and share with you some of our efforts to building a community to really propel the medical science and genomic science towards building an accurate and most relevant human cell atlas for the future. So I just want to start off, and it's I think it is an important reiteration to all the members of this meeting, as well as the HCA community, is to really reinstate the idea of building and creating this comprehensive reference map of the human cell types. And really the fundamental units are the human cells in life, because we want to really interrogate one of these individual cells to understand, diagnose, monitor, and treat health and disease. And there's been various analogies, Google Maps, and here we're showing the analogy of periodic table. Really the idea is to comprehensively tease apart individual cell types so that we can understand what are the molecular makeup of each cell type, but what are the relationships from one cell type to the other in the context of bigger tissues or organs or the human physiology. So really, with the advancements of single-cell genomics, I think we have entered into this realm of single-cell profiling, and we're very close to achieving that goal. But this would not have been possible through really long time and efforts by various groups around the world. Since the launch in 2016 in London, there has been various meetings, not only in general meetings, but multiple HCA Asia meetings that Alistair also had mentioned. There's been also a launch of biological networks where there are seminars and series of talks that we will highly encourage everyone in this group to also participate and even be invited to speak. This is a current status of the countries that are part of the Global Human Cell Atlas community, where 79 countries, over 1,000 institutes, and 2,000 members are participating. And I just took a notice of this region here that I've highlighted with this dashed line. Collectively in Asia, we have over 300 members represented in this global community. And this is not a small number, and I'm really excited and very empowered by this active participation and interest of the scientists here in the region. And we really look forward to working together and building a greater and stronger community in the region. As I mentioned earlier, the Human Cell Atlas has set out to create the biological networks. So far, there are 18 Human Cell Atlas networks, and you can see the various tissues or the organs that are led by various coordinators. And the philosophy behind this network is to kind of divide and conquer this Human Cell Atlas by bringing together the experts of individual tissues or organs to really create the most comprehensive yet accurate atlas with the biological expertise. And so I would also highly encourage you, if you are an expert in any of these tissues, to contact these coordinators and actively engage in either computationally or sample provisions or technology in order that we can create these atlases for individual tissues. And ultimately, of course, we will coordinate these biological networks into a one global human cell atlas that we hope to achieve in the near future. But I just want to highlight this one biological network led by Shyam Prabhakar, who will speak after me, and Sarah Tishkoff and Jimmy Yi, where it's a component that is very important for us in HCA Asia, but also globally, to really understand the genetic diversity that are so present in this world. And I just really encourage you, whether you are experts in these biological tissues, yet have access to various tissues from different regions, to contact Shyam, Sarah, or Jimmy to dive into the genetic diversity question of the human cell atlas. This is a quick view from the atlas, and this has been shown also at the general meeting by Sarah as well as Aviv. But this is just to highlight some quick updates on what the various research areas. These are the 18 biological networks that I've mentioned. But also we have collected over samples from 7,000 donors from 35,000 samples. But surprisingly, we have now, as a community as a whole, have generated 54 million single cell data points, and 11 million addition that are related to COVID-19 samples. So these are kind of quick breakdown and the view of the atlas, just to highlight. Here in the eye, over 2 million cells have been profiled. In the colon, 5.2 million cells, over 1,000 samples from 400 donors, as well as reproductive system. And we can go further into developmental system. We have 7.5 million cells, as well as the brain tissues, as well as blood samples as well. So these are data that's been generated by the community at large, and this is growing by the minute. And as you can see, over the last 3-4 years, there has been a rapid increase in not only on the samples, but number of individuals that we are profiling, and obviously the number of cells that we have collected. And we are definitely looking forward to expanding this in order that we can really understand the level of variations that we see across samples and across individuals, so that we can accurately map the healthy reference of the human cell atlas. So one of the big efforts of the human cell atlas is to create a data portal. And here is a snapshot. This is probably already a bit outdated from what I've seen yesterday. It's already over 20 million cells have been deposited into this data portal. And you are freely able to navigate and filter based on gender to age, sample organ technologies and methods, and to download these individual data for your perusal and for your analysis. So it's very powerful, and it is growing, and there's strong efforts to integrate additional data in the future that may be both through either full access or managed access. So I just want to go back to this mission that we have stated in 2016 when we launched the Human Cell Atlas. To create this human reference map, we start asking, how can we best create the most relevant reference? And this quotation relevant really pertains to the idea that how can we create a reference that is most appropriate for every individual, or at least in the local regional population that we see across the world. And this has been very much prevalent in the past few years, because many of the genomic data that have been generated have been largely biased towards European descendants, and that these databases, Genome AD or GTEx, have very few representation from Asia, where more than 50% of the world population are from Asian countries. So after talking to various members and communities, there are two fundamentally inhibitory factors which are one, limited access to many of these advanced technologies and the know-hows on how to analyze this large genomic data set. And then the second barriers to entry was really largely the funding, mostly from low-income to middle-income countries that have limited funding sources to produce a large number of sequencing data. So to overcome a lot of these challenges, we have launched and have been having this annual HCA Asia meetings. And really the goal and the purpose really is to create awareness towards equitable participation and representation across Asia in order that we can really highlight key science and technologies from the region, but also to identify and tackle areas of societal needs and challenges, and to really bridge the gap that really creates the barrier to entry to these kind of technologies. So 2017, we had our first annual HCA Asia meeting, which had maybe 30 or 40 members, and that was hosted by Piero and myself. And then in 2018, we had our second HCA meeting in Jeju Island, South Korea, hosted by Sun-Young Kim and Eun-Young Park. And then in November 2019, we had another meeting hosted by Shayan Prabhakar. And last year, we had our first virtual meeting hosted by Goji Go. And I just want to highlight that every year, there's been an active and increased participation and just really very strong community and dynamic that has been building. And hopefully, we will see some of the key highlights that have been generated through these HCA Asia meetings in the next 2 days. So we are here in the 5th meeting hosted by Alistair in Australia, and we're very excited to discuss what are some of the latest technologies and how we can further embolden and strengthen our community. I just want to keep mentioning the word community for the past few minutes, and this really has been one of the key components and key drives for my personal research experience here in Japan. And I just want to share with you some of the examples and my experience, perhaps a case study of how we have built the medical research community in Japan. And perhaps this could be used not only in your own countries, but also in Asia to bring together expertise of different domains to tackle a common issue. So in Japan, we have a wealth of medical universities and biobanks with exceptional, really high quality clinical data and access to human samples. But one of the key barriers was that there's been a huge gap between clinical community and the genomic community, that there was a very small or little exchange of knowledge in how to analyze sequencing data or how to process and even make next generation sequencing libraries, but even further, how to analyze this kind of big data and analytical tools. So this was really an opportune time for us to bridge the gap between medicine and genomics in Japan, and ultimately to integrate and disseminate and to ultimately advance the medical science here in Japan. But to achieve this, Weekend has been reaching out to various medical institutions in Japan through giving talks and really having roadshows, visiting various universities from University of Tokyo to even Keio University, Osaka University, and so on, to pitch the idea and really provide the power of single-cell genomics to many clinical researchers as well as doctors who see patients every day. And the idea was to create a win-win scenario for the community. And what I mean by that is many of these clinical researchers and doctors, their main interest is specialized in various disease and tissues, and they want to understand the disease and tissue in a deeper level. So inevitably and inherently, they are very curious about the single-cell genomic data. However, they do not have the knowledge or the technologies to do this. So we as a community then generates individual data for the PIs or the clinical researchers for them to investigate and dive deep into. And a lot of these collaborations and work have been, of course, supported by many of the IRB and HCA Japan and HCA Weekend coordinator Yoshinori Ando-san. And the idea is that we generate data and we feed back to individual or clinical scientists. But in return, as a genomic and genomic interest, we take these single cell data from various tissues and we integrate to understand what is the cell type specific expression or what are the regulatory motifs, and how can we further integrate human genetics to understand cis regulatory networks. So this really has been a very effective tool to not only provide single cell data to individual clinicians and biologists, but also use this integrated data to understand population genetics and gene regulation at large. So we've gone through various stages of engagements and IRBs and forming research agreements. It is a lot of work, but it is definitely worth exploring since we can really access to very rich human clinical data. And we go through a very rigorous and standardized QC of tissue generation, tissue dissociation to library productions to data QC. And then that can lead to various paper writing and grants and so on. So these are some of the examples that the data that we provide to individual clinicians or research scientists who are not familiar with single cell data. And these are standard packages that are currently available. And we also use and leverage the Cell by Gene platform created by CZI. And this particularly the new edition called Visualization and Plugin has been extremely taken up with enthusiasm by a lot of our collaborators who are then able to use this plugin to generate their own figures. Really has been very effective and a great way to engage with scientists who are not familiar with single cell data. So here are just quick examples of some of the clinical researchers that we deal with. They're investigating atopic dermatitis and using both single cell data and Visium data. We have doctor working on schizophrenia, as well as gut immunity, as well as salivary gland, looking at Jorgen's syndrome. So these are just key examples of how we can engage in various clinical samples as well at the same time address a very specific disease that are interest to many of the clinical scientists. But what in return is that what we are able to do with this rigorous standardization is to integrate the data to understand large-scale genomic information. And this is a field that Weekend has been pioneering for several years, largely to understand how gene regulation takes place through promoter and enhancer regulation. And we do this by applying the 5' RNA-seq technology. But this is not new, and you see Alistair Forrest, who have led the Phantom project to understand the promoter of various tissues in bulk, and Alvin Satterlin, who have looked at enhancer RNAs across multiple tissues to understand enhancer activities. So taking this idea, we have developed a single cell-based 5' cage technology to understand and to identify enhancer activities. We are now able to employ the same technology using 10x genomics to identify promoters and enhancers that are upstream of the gene or the downstream. So here, we have developed a software mainly led by Dr. Chung Chao Hong, who's a PI and a good collaborator at Weekend. We're able to identify a genuine transcriptional start site at the 5' end in order to clearly define the promoter regions. So I highly encourage you, if you're using 5' RNA-seq technology, to browse through this tool to help you identify a genuine TSS. And here we are able to identify a very clear indication of a cage peak or the TSS peak defined by single cells here, as well as enhancer RNAs, which tend to be bidirectional with plus and minus strand. And here you can see the individual rows are from single cell data, highlighting that single cell expresses either one direction, but not the other. So it's actually a unidirectional pattern that is indication of enhancers at the single cell resolution. So we use these kind of definitions to define transcriptional start sites, and we call them cis regulatory elements. So these are, we use this information to benchmark the transcribed cis regulatory elements compared to the accessible or the ATAC-seq defined cis regulatory elements. And we show a very strong correlation between the two, where whether you use the transcribed or the accessible, you do find a very good, strong commonality. But one additional factor that we are able to gain through transcribed cis regulatory elements is the dynamic range. Because this is based on gene expression, we are able to understand the difference between the activation and depression, where accessibly defined cis regulatory elements are either present or absent. So here's an example of one transcribed CRE that it shifts from high to low upon stimulation, where you have low to high, but in an ATAC-seq, you will not be able to tease apart these dynamic regulation by ATAC-seq. So we have used this type of CRE definition to understand cis regulation. And here is a classical example of PTGR4, which has been known to involve in Crohn's disease. But we are able to show that it's not the protein coding gene, but then the enhancer regions that are upstream of this that are full of SNPs that are associated to Crohn's disease. And when you zoom in closer, we do get a very strong transcribed transcription of enhancer RNAs that we can utilize to understand that this co-expression is linked to the promoter of PTGR4. So we are using this to understand the difference between a ATAC-seq defined CREs versus the transcribed CREs. So here is an example that if you have an open chromatin defined by ATAC-seq, but it has no evidence of transcription, we tend to find a very poor enrichment of a heritability that are associated to immunologic disease traits. So for example, ulcerative colitis, if you have an ATAC-seq with no transcription, have a very poor enrichment of heritability, where if you have a ATAC-seq signal with a strong evidence of transcription, have much more enrichment of that particular disease traits. So we reason that many of the ATAC-seq signals could not be enhancers, that could also be cohesions or other structural components, and that these actively transcribed regions of the ATAC-seq may indicate a strong functional implications for a lot of these disease traits. So just to summarize all of our efforts in the single cell community in Japan, here we have collected over 500,000 single cell data highlighted by tissue, but also by major cell types. And we can now dive deeper into understanding the enrichment of trait heritability for individual tissues and understand what are the potential disease traits that are associated to promoter enhancer regulation. So here is an example of one disease, coronary artery disease. And when we look at endothelial cells, we see a strong enrichment of heritability here in this part of the endothelial cells, but very low enrichment in this part of the endothelial cells. And if you look back here on the left and start color coding based on the tissue of origin, the endothelial cells from the heart and muscles tend to be highly enriched for coronary artery disease, but not by other tissues. As a control, when you look at the heritability of enrichment of inflammatory bowel disease, or IBD, you can see that it's quite scattered apart, which suggests not really tissue specific, where coronary artery disease is so. So in summary, I just wanted to highlight that we do recognize that there's still gaps between clinical and genomic research. And we have established this win-win scenario where we can actually provide the genomic technologies to clinical communities, and that these clinical researchers can take these technologies and use tools like cell by gene and other softwares to investigate the disease of interest. But at the same time, we are able to integrate all of these clinical data to generate a database that will help us, that are helping us to expand our knowledge in human genetics and to understand this cis regulatory network. So we are continuing this type of efforts to generate data and share our data to advance science and medicine. So I just want to close here by saying, for the next 2 days, these are very common questions that we could be asking, such as, what can I gain from the HCA Asia meeting? And, especially if you're a student or new to the field, I think this is an appropriate question. But I think the question that we could also be asking, especially if you're a PI or an expert in certain technologies, to ask what kind of technologies or how can my expertise contribute to HCA Asia community and create this win-win scenario that will not only help my own research, but the community at large. So hopefully this could be a reoccurring question that we could be asking throughout today and tomorrow, and hopefully come out in the end with a very strong project proposals or grant proposals that we can build upon. So with that, I would like to close by acknowledging all the people in the single cell medical network in Japan, a lot of them who have contributed to the sample provisions and their help in getting the ethics and data production and funding, as well as the Human Cell Atlas CZI for their support. And also I just want to highlight Chung-Cha Hon, who have really been key drivers of developing a lot of these software tools, and Aki Minoda, Takeya Kasukawa for data integration, Piero Caninci. And also want to acknowledge Jonathan Moody, who's been the bioinformatician, a postdoc who's been driving out the analysis as well. Yoshinari Ando for coordinating a lot of these efforts, Tsukasa Kono, Miki Kojima, and Ikuko Koya for other technical support. So with that, thank you for your time and happy to take any questions.
Great, thank you for that, Jay. Just a reminder to everyone online, if you want to send questions, please do those through the platform, and we'll ask them for you. So, Jay, I mean, you correctly identified sort of two of the barriers. One was funding and one was ethics. And I'm interested to know, with the single cell medical network that you just mentioned at the very end, so each of the hospitals are providing samples. Who's paying for where's the funding coming to pay for each of these? Because lots of hospitals want to collaborate, but each of these assays are expensive. So
yeah, so we have mixed bags. So we have clinical scientists who have their own funding, yet they just don't know yet how to use it to the fullest in terms of data production. So in that case, we help them spend their budget and generate the data that they need. I would say in Japan, we've been quite fortunate that many of the collaborators have had their own funding to generate. But there are a few exceptions that were not able to fund themselves. In that case, if the research aim aligns well with what we are doing here at RIKEN, or some of the key biological themes that are active in RIKEN, and there's good alignment, my lab or some of the labs here at RIKEN will support that data production.
Cool. And regarding the ethics, so you're dealing with lots of different hospitals, and some of these samples may have been collected previously, some will be collected prospectively. So is it a case-by-case basis there as well?
Yeah, so many of these clinical researchers already have ethics, except they don't have the clause on genomics, right? So many of them have ethics to do research on. So what we do is we ensure that their application is updated from their existing ethics that includes genomics profiling. And on our side, we already have a common template that we use that already has been accepted. So what has helped is that we created an ethics, or the IRB approved ethics in Weekend as a common template that can be utilized to various institutions in Japan. And we were able to do that because we created our own kind of local consortium that this ethics falls under. So that's probably a good way if you're planning to collaborate with many different institutions to have one template under a one project and work with individual IRBs, but mainly has been just updating one or two lines in the application that adds the term genomics profiling. And that has been quite effective. It's time consuming, but it has been effective.
Cool. And I guess one more question, because there's none come through yet. The ability to share data internationally. So I know Sham's going to talk about ADA in a minute, but with your ethics, what are you allowed to share? Is it raw sequence level data, or is it behind something like dbGaP, etc.?
Yeah, so we initially, when we engage with individual collaborators that we want to open the data, and that we also, during the IRB review process, we explicitly say we want to open the data. And so far, generally has been quite positive from our side that has been allowed. I think there's been I think that is not going to change so far. There's been, in fact, further simplification of how we can release the genomic data going forward by even having single IRBs instead of having two IRBs from two different institutions. So I feel, at least in my opinion, that the trend is that there's a drive to further simplify, to help researchers to collect samples, but also to disseminate data as quickly as possible. So hopefully that trend will continue across, not only in Japan, but across the worldwide.
Okay, cool. Thanks for that, Jay. I think in the interest of time, we need to move on to the next speaker. There is a question from Christine, but maybe you and Sham can answer that at the end of Sham's presentation. Okay. So thanks, Jay.
Thank you.